People often say the human brain is the most complex object in the universe. And when you think about it, with its 86 billion neurons, each one making thousands of connections, it's hard to argue. But what's truly mind-boggling isn't just the scale. It's the incredible diversity. Neurons are not all the same. So how can a scientist possibly pick out and study just one specific type of cell in this huge tangled network? Well, the answer lies in learning how to speak their language, their genetic language. Just take a look at these pyramidal neurons right here. Even inside a single class of neuron, their shapes, their morphologies are just wildly different. A cell from layer V of the cortex looks nothing like one from the hippocampus. And this very diversity, in their shape, their molecular makeup, their electrical signals, well, that's exactly where the brain's massive computational power comes from. But it's also what makes studying them an absolutely monumental challenge. So in this explainer, we're going to break down the revolutionary technology that makes this all possible. We'll kick things off with the big picture problem, then we'll get into what a transgene actually is. From there, we'll see how these genetic tools are designed and controlled, check out some really powerful applications, and wrap up by talking about their massive impact on the entire field of neuroscience. Okay, so think about it like this. To understand any complex machine, you have to be able to take it apart and test its individual components, right? The brain is no different. The fundamental problem that transgenes solve is giving scientists a tool to isolate and even manipulate specific groups of neurons inside that dense, tangled-up network. So what does this let us do? What's the payoff? Well, by hijacking the unique genes of certain cells, we can literally label them to make them visible, which then lets us trace out all their intricate connections. We can even take it a step further and record the electrical or chemical signals specifically from those labeled cells. And what's even crazier, we can actively manipulate their activity. We can turn them on or off to see how that messes with the circuit and even an animal's behavior. In some cases, you can even just get rid of them completely to see what happens when they're gone. All of these incredible abilities are made possible by one tool, the transgene. So let's get a really precise definition of what we're talking about here. A transgene is, pretty simply, a piece of foreign DNA. It's a gene, or maybe a set of genes, that scientists have designed from scratch and then artificially inserted into an organism's own genetic code, its genome. The whole point is to give specific cells a brand new ability they wouldn't have otherwise, like the ability to glow green or maybe respond to a flash of light. Now, there are basically two main ways to get this foreign DNA into an organism. You can do it right at the very beginning by putting it into the germline cells, the sperm or the egg. If you do that, you create a change that's heritable. It's going to be in every single cell of that organism, and it'll get passed down to its kids. The other way is a somatic introduction. This is where you deliver the transgene locally to a specific spot in a fully developed animal, targeting cells that aren't reproductive. That change is not heritable, and it stays confined right there in the cells you targeted. Now, for a transgene to actually work, it has to be designed super carefully. You can think of this chunk of DNA as a construct. It's like a genetic instruction manual that tells the cell what to make, and just as importantly, when and where to make it. So let's break down that instruction manual. A basic construct has three essential parts. First, the promoter. Think of this as both the on switch and the address label. It's a sequence of DNA that is only recognized by the machinery in a very specific cell type. So if you use a promoter that's only active in, say, dopamine neurons, your transgene will only get turned on inside those cells. Next up is the effector gene. This is the payload, the gene that actually does something. And finally, you have a terminator sequence, which just tells the cell's machinery, okay, stop reading here. And the choice of that effector gene? Well, it completely depends on what question you're trying to answer. It could be a simple reporter protein, like green fluorescent protein, or GFP, which literally just makes the cell glow. Or it could be a light-sensitive channel, like channel rhodopsin, which lets you control the cell's activity with flashes of light. It could be an imaging protein, like a calcium indicator, that lets you watch the cell's activity in real time. Or it could even be something called a transcriptional activator, a protein that acts like a master switch to turn a bunch of other genes on. And this just brilliantly shows you the power of a simple reporter. By sticking GFP under the control of a neuron-specific promoter, scientists can light up entire nervous systems, like you see in the zebrafish, or highlight specific groups of cells with just stunning clarity, like these Purkinje cells in the mouse cerebellum. All of a sudden, you can see the beautiful, intricate structure of cells that were totally invisible before. Okay, so a cell-specific promoter gives us incredible spatial control, the where. But what if we also want temporal control? What if we want to control the when? To get that, neuroscientists have cooked up even more sophisticated multi-part systems. And this is where things get really clever. Instead of having everything in one package, scientists use what are called binary or two-part systems. So if you look on the left of this diagram, the first transgene, the driver, has our cell-specific promoter. But instead of making our final protein, it makes a special kind of switch. The second transgene on the right, the responder, has our gene of interest, but its promoter only works when it gets flipped by the switch from the driver. And here's the key. Our final gene only gets expressed in cells that have both of those pieces. A classic example of this is the Cre-Lox system. What you do is breed two different transgenic mice. The first one, the Cre mouse, expresses an enzyme called Cre recombinase. You can think of it like a pair of molecular scissors, but only in a specific cell type. The second mouse, the Lox-P mouse, has our target gene, but it's blocked by a stop sign that's surrounded by special tags called Lox-P sites. So, in their babies that inherit both transgenes, those Cre scissors are only made in the right cell type. And in there, they find those Lox-P tags and physically snip out the blocker, which finally allows our gene to be expressed. It's a way to get incredibly precise spatial control. Now, to get that temporal control, the when, we can use inducible systems, like the TET system. In what's called the TET-off version, a special activator protein is always on, making your gene of interest. But if you give the animal an antibiotic, like doxycycline, it grabs onto the activator and shuts it down. The TET-on system is just the reverse. The gene starts out off and only turns on when doxycycline is around. This gives researchers a reversible switch to turn genes on or off at any point in an animal's life, just by changing what's in its food or water. So, with this incredibly sophisticated genetic toolkit, what are researchers actually doing to figure out how the brain works? Let's walk through a concrete example. What you're seeing here is a simplified circuit diagram from the hippocampus, a part of the brain that's absolutely critical for memory. It looks complicated because, well, it is. But notice all those different colored cells, PVBC, OLM, VIP-LRP. These are all different subtypes of inhibitory interneurons, and each one has a very unique job. Before transgenes, telling them apart would have been nearly impossible, but with transgenes, we can target each one of them specifically. And this is how that specificity leads to real discoveries. A researcher can use a transgenic line where only one type of cell, like these parvalbumin-expressing basket cells, is made to glow. Now, when they look under a microscope, they can easily find that glowing cell, patch on with a tiny electrode, and record its unique electrical fingerprint. By doing this over and over for every different interneuron type, scientists can literally build a complete parts list for the circuit, linking a cell's molecular identity to its specific shape and its unique job. You know, the development of these transgenic tools has done so much more than just help us answer specific questions. It has fundamentally changed the entire way neuroscience is done. By creating standardized mouse lines and genetic constructs, this technology has created a shared language. A lab in California can study the exact same genetically defined cell type as a lab in Germany, and they can be totally confident they're talking about the same thing. This has really enabled this systematic, worldwide effort to classify and understand every single cell type in the brain. But really, the most profound impact is how it completely changed the kinds of questions scientists could even ask. For decades, neuroscience was mostly correlational. You could see that when a brain area was active, a certain behavior happened. But you could never really prove that one caused the other. Transgenes changed all of that. For the very first time, scientists could take control. They could turn a specific set of neurons on and ask, does this cause the behavior? Or turn them off and ask, does this prevent the behavior? And that is the critical leap from just seeing a correlation to actually establishing causation, which is the absolute gold standard in all of science. By giving us the power to deconstruct the brain's complexity piece by piece, cell by cell, transgenic technology has blown open doors to frontiers we couldn't have imagined before. And it leaves us with this final, really tantalizing question. As we assemble this incredibly detailed parts list of the brain, how does understanding the function of each tiny component bring us closer to understanding the big, emergent properties of the mind, like perception, memory, and maybe even consciousness itself?